Assalamualaikum and a very good morning. I bid to the panels and my friends. My name is Aishah and I'm going to present my final year project on a review on immunotherapies for melanoma. Melanoma is a skin type cancer that involves mutation in BRAF oncogene. It alters protein that regulates cell growth, thus causing melanoma to grow aggressively. It is a heterogeneous tumor with low cure rate that develops through malignant transformation of melanocytes. Currently, there are lack of effective conventional treatments for melanoma. Therefore, immunotherapies are introduced to augment immune system by modulating tumor microenvironment. The immune cells will attack cancer cells that will then improve survival rate of patients. Two problem statements identified, which are the resistance of cancer cells toward conventional treatments. Also, conventional treatments can directly damage DNA. This study is significant to further understand the abilities of immunotherapies as alternatives for conventional treatments in treating melanoma. Also, it helps researchers in identifying promising and effective treatments for melanoma. The research questions are listed as follows. What are immunotherapies? What are the differences between conventional treatments and immunotherapies for melanoma? What are the current and potential immunotherapies that can treat melanoma? Two objectives presented in this study. First, to compare between conventional treatments and immunotherapies in treating melanoma. Second, to review current and potential immunotherapies that can treat melanoma. Now, moving on to the introduction to immunotherapies. Immunotherapies are cancer treatments that modulate immune system to recognize and attack cancer cells. They can also boost immune cells to remove cancer. They form anti-cancer reaction by increasing quantity of effector cells and forming soluble mediators that allow tumor-killing condition to take place. For melanoma, immunotherapies are preferred over conventional treatments. This is because they can recognize and attack cancer cells before the host is harmed. They have high specificity which they attack only cancerous tissue. They possess higher effectiveness. In contrast, conventional treatments can evoke the redevelopment of cancer cells. They are non-specific which they cannot differentiate between cancerous tissue and normal tissue. They have high toxicity. One of the current immunotherapies that can treat melanoma is immune checkpoint inhibitors. Cancer cells can escape detection by binding to immune checkpoint proteins as receptor sites. They can be programmed cell death 1, PD1. Meanwhile, the ligand for PD1 will be programmed death ligand 1 or 2, PDL1 or PDL2. When immune checkpoint proteins bind to their ligands, an off signal is sent to T cells to protect cancer cells. Therefore, inhibition of immune checkpoint proteins must be initiated. Besides, there is adoptive cell therapy, chimeric antigen receptor CAR T cell therapy. Based on the figure, T cells are utilized to attack cancer cells. T cells are first isolated from the patient and being modified to produce a protein named CAR. They will be then grown in a large quantity and reintroduced back into the patient to kill those cancer cells. In addition, oncolytic virus therapy allows viruses to replicate themselves. This method utilizes modified viruses that are non-virulent to normal cells. They will then take over protein factories of cancer cells and induce inflammation as cell death occurs. The process will then stimulate dendritic cells and T lymphocytes. Antigens are released as lysis occurs. One of the potential immunotherapies that can treat melanoma is bacterial-based immunotherapy. It can utilize non-pathogenic bacteria as they can regress tumor. A recent study found that bacteria are effective as they are cytotoxic and can inhibit growth of cancer cells. This method can regulate immune system components such as NK cells, CD4 and CD8 T cells. For example, there are bifidobacterium species which are part of normal microflora that exert probiotic properties. They can enhance the function of dendritic cells and can improve antigen-specific CD8 T cells. This method can be joined with PD-L1 antibody therapy for greater effectiveness. To conclude, immunotherapy is a cancer treatment that involves immune system to target and kill cancer cells. It is beneficial as it has high specificity and has high effectiveness. Since current immunotherapies for melanoma is limited, it is necessary to find potential immunotherapies that can treat melanoma effectively. Through this, people whose current treatments have failed have higher chances to recover. Currently, there are lack of research and data regarding the potential immunotherapies to treat melanoma. Therefore, it is recommended that further research is conducted to solve any problems or issues arise. These are the references used for this presentation. That's all from me. Thank you.